So in part 2 of this set of lectures, we're going to talk about some experiments in phages. So Felix de Herelle is the microbiologist who's widely credited with discovering bacteriophages. So in 1910, he had actually been studying controlling insect pests of crops using bacteria. He discovered some bacteria that killed locusts, which are those grasshopper bugs that swarm all over crops. And he had been growing these bacteria in his lab in order to spread on those crops to stop the swarms. He noticed on these plates that some of those plates had these round clear spots, and he asked, what created this clearing? Frederick Twort is also a microbiologist who around the same time had independently discovered some of these round clear spots on his plates of pathogenic Staphylococcus. But Twort's research was unfortunately interrupted by World War I. But these two men are often credited as co-discovering bacteriophages. So de Herelle had been able to understand more about these bacteriophages by doing an experiment. Okay. So de Herelle took his sterile swab from his microbiology lab, poked it into the round clear spot, and when he spread that on a plate without bacteria, nothing grew on that plate. But if he took a new plate and spread some bacteria on it, took a little sample from that spot, he found that there were new clear spots. So he wanted to know, is there something that is transferable between these clear spots that causes the reappearance of clear spots? So what he did was this. De Herelle examined the spots in a microscope and he couldn't see anything. He was able to see bacteria on his microscope, but he couldn't see anything in those clear spots. So de Herelle used an instrument called a Chamberlain filter. So Chamberlain filters had been invented to remove bacteria from liquid. They were often used around 1900 to prevent the spread of cholera, which commonly infected many water sources. So de Herelle filtered his spots through the Chamberlain filter, which he knew would remove any bacteria from the sample. He filtered his liquid from the spot and spread it on the plate. What do you guess is the result? So I can tell you that the result was that he still saw spots. And this was consistent with some recent work that had discovered things that cause disease that were even smaller than bacteria, things that were viruses. And so de Herelle hypothesized that there was a virus or a smaller than bacteria particle that was causing these diseases, this clearing, this death of the bacteria. So we now know that these are plaques. They're little clearings on the plate caused by bacteria being killed by the phage viruses. Right? So we're going to use plaque assays extensively in this course, but one critical skill to think about is how do you determine that a clear spot on a plate is actually the result of a phage? Right? You'll notice as you go along that, such as this, the result that we see on the right here. So on the right we have some top agar that was put on the plate that just wasn't spread carefully. And instead of forming a nice even layer of bacterial growth on the plate, there's some big gaps where things just weren't touched by the soft agar. So this is a clearing on the plate, but it's not caused by bacteriophages, it's caused by sloppy experimental technique. So when we do a plaque assay, we have to determine, is a spot on the plate the result of a phage? Is it a bubble in the agar? Is it at somewhere we accidentally poked the plate with our pipette? We want to be able to think, how can we determine if that plaque assay, if that spot we see on the plate is actually the result of a phage? So think about, what are some other reasons bacteria might be risking for a spot? So we talked about poking your plate, some bubbles. But if it is a phage, how would you expect phages to behave? Look in the phage discovery guide and determine a protocol that might be useful. We'll talk about this in Tuesday's class. So This is the experimental design for the entire Phages course, and we're going to be working through this process throughout the semester. So you're going to start by collecting soil samples, which you'll be doing here now. Right? But once we collect the soil sample, we want to know, are there phages in it? So we'll bring it back to the lab and perform a plaque assay to determine if we can get plaques formed from that plate. If we see plaques, we want to determine if they're actually caused by phages. And so we're going to pick them into phage buffer where we can store phages for a short period of time. And then perform an assay that we call a spot test. We're actually going to determine whether or not that plaque is the result of a phage. We then want to demonstrate that the phages we find are just one type of phage, because we actually know that there is a huge diversity of phages in the environment. And so we want to be working with just one phage so we can characterize it specifically. We're going to perform a number of rounds of what we call purification. So we're going to repeatedly dilute the phage so that we can get single plaques in a plate, pick phage from a single plaque, and observe that the morphology of those plaques we get is the same in every single experiment. Once we're confident we're working with a single plaque, then we can scale up. We can make a large lysate, or a sample that has a huge number of phages. We're going to be trying to get a lysate that has over 5 billion phages, or plaque-forming units per milliliter. And we need to be able to calculate how we get this number. If we can get a high titer lysate, we're able to do things like archive the phage, where we make an ultra-cold stock of the phage that can be stored for a long period of time, right? And we can enter that sample into the Phages DB actinobacteriophage database. Then we can study this phage in more detail. We can do DNA extractions and characterize that DNA using a number of molecular methods. And we can also do additional experiments to figure out how that phage behaves. So the ultimate goal is to find a phage, develop enough bacteria, right? So develop enough DNA from that sample. Sorry, not bacteria. We're going to develop DNA from the phage sample so that we can get that phage DNA sequenced by the University of Pittsburgh and determine the genome of our new phage and see if it actually contributes something novel to the genetics, the diversity of phages in the environment. So we're going to really build evidence for this through a number of steps. So we're going to want to make sure that we can find a sample where we can isolate a phage. We want to do that purification where we can determine only one phage is in that sample. Then we want to be able to count the number of phages in your sample. We want to be able to extract DNA from those samples and make sure that DNA that you extracted looks like phage genome DNA and is not contaminants from the bacteria that we're using as hosts. We're going to be doing a number of things throughout the semester to build evidence for each one of these things, but today we're going to talk about how you count the number of phages in your sample.